Hello everyone, welcome to our YouTube platform Concept and Fact of Life Sciences. Today in this video we are going to discuss about the enzymes. For enzymes there is a statement that all enzymes they are protein but all protein they are not enzymes. This is a very much true statement, like 99% of the enzymes they are protein. Why 99? Because we are having the exceptions in the form of ribozymes. These ribozymes, these are the catalytical RNA. So, all enzymes with the exception of ribozymes are protein. Now, for enzymes, there is a Gibbs free energy for activation and it is denoted by delta G hash. This reaction, it is representing the photosynthesis in which there is a conversion of carbon dioxide and water into the glucose. In this direction, the reaction is energetically unfavorable, which requires the investment of energy in the form of sunlight. But in the reverse direction, the reaction is energetically favorable. But the glucose will not convert into the CO2 and H2O spontaneously. First, we have to drive this glucose into its transition state for the conversion of this glucose into the CO2 and H2O. Now, the energy which is required to drive the molecule into its transition state is known as the activation energy. Lower the activation energy means lesser the energy which we require to drive the molecule into its transition state, more is the rate of reaction. So the rate of reaction, it is inversely proportional to the activation energy. Now this activation energy, it gives free energy for activation. It tells us about the rate of reaction. But the reaction, it will occur or not, it depends upon the concentration of product and the substrates. We try to understand this activation energy and the effect of enzyme on this activation energy. First, there is a conversion of this substrate into the product. It requires this much of activation energy. While in the presence of enzyme, the conversion, it requires only this much of activation energy. So we can say that the enzymes, they decreases the activation energy and thus they increases the rate of reaction. The order of reaction, it is the effect of reactant on the rate of reaction. If the rate of reaction, it depends upon the concentration of single substrate. If it depends upon the concentration of single substrate, then it is known as first order reaction. If it depends upon the concentration of two substrates, then it is called as the second order reaction. And if the rate of reaction, it is independent of the substrate concentration, then it is known as the zero order reaction. Now, there are some characteristics of the enzymes. Number one, they alter the rate of reaction. As we already studied that the enzymes, they decreases the activation energy. They decreases the activation energy and thus they alter the rate of reaction. So we can say the enzyme, by decreasing the activation energy, they alters the rate of reaction. They stabilize the transition state of substrate. Enzymes, after binding to the substrates, they makes conformational change in the substrate and stabilize them into a transition state. So enzyme, after binding to the substrate, they stabilizes the substrate into their transition state by making the conformational change in them. Third, they do not disturb the equilibrium. The enzymes, as we can observe over here, they decreases the activation energy for both forward rate of reaction as well as for the reverse rate of reaction or the conversion of product into the substrate. So they do not disturb the equilibrium. Next, factor affecting the enzymatic activity, and there are two major factors. One is temperature, other is pH. If we talk about the temperature, it is directly proportional to the enzymatic activity. As we increase the temperature, the enzymatic activity will also increase, or the rate of reaction will also increase, but only to a particular extent. Why? Because at higher temperature, as we all know that the enzymes, they are made up of protein, or they are protein. So at higher temperature, the enzyme, it will get denatured. So with temperature, the rate of reaction increases, but to a particular extent, because at higher temperature, the enzyme, they will get denatured, because the enzymes, they are made up of protein, and the protein, they have their functional tertiary and secondary structure, and on higher temperature, then the permissible limit, the protein secondary and tertiary structure, they will get denatured, which hinders the functionality of enzymes. Second is the pH. All enzymes, they functions at the optimum pH. Because on optimum pH, the conformation of enzyme is such that it will readily recognize the substrate. We can take the example of acid hydrolysis, which functions within the lysosomes, and if they comes out into the cytoplasm where the pH is around 7, they are not able to carry out their function. So all the enzymes, they will function at the optimum pH. Now there is a classification of enzymes. The Enzyme Commission classified the enzymes into 6 different categories. Number 1 is the oxidoreductases, which includes oxidases and dehydrogenases, which carries out the oxidation and reduction reaction. Next is the transferases. The transferases in biological system, the best example for transferases is the kinases, and these kinases, they require two substrate. We can take the example of enzyme that is called as the glucokinase. Now this glucokinase, the function of this glucokinase is to convert the glucose into the glucose 6-phosphate. Now there is an addition of phosphate onto the glucose 6 position, and for this they require the energy in the form of ATP. The conversion of ATP into the ADP plus inorganic phosphate. And this inorganic phosphate, it will be transferred to the glucose for its conversion into the glucose 6-phosphate. So these glucokinase, they require two substrate. One substrate is the ATP, while other substrate is the glucose. It transfers the phosphate group from one substrate to the another substrate. So these are the transferases with the example of kinases. Next is the hydrolases. These includes the digestive enzymes. And these digestive enzymes, they generally requires the water molecule to break the other molecule. For example, the amylases, which carries out the breakdown of starch. We are having lipases, which are involved in the fat breakdown. Next category is the lyases. These lyases, they do not require, or they carry out the breakdown of molecule in the absence of water molecule, which includes the aldolases and fumarases. Aldolases in the glycolysis, we can say it carry out the breakdown of fructose 1,6-phosphate into the glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So it carries out the breakdown in the absence of water. That's why they are belonging to the category of lyase, EC number 4. Now fifth number is of isomerase. They converts one isomeric form. For example, the glucose 6-phosphate conversion into the fructose 6-phosphate during the process of glycolysis by the enzyme which is called as mutase or hexoisomerase converts a glucose that is the 6 carbon aldehyde sugar into a fructose that is a 6 carbon ketone sugar, one isomeric form to the other isomeric form. The enzyme is glucoisomerase. So these are the isomerases which converts one isomeric form into the another isomeric form. Examples are mutases, epimerases, etc. Sixth category we are having ligase, and a classical example of this ligase, it includes the amino acyl tRNA synthetase. Now the function of this amino acyl tRNA synthetase is to add the amino acid onto the tRNA as per the anticodon sequence to convert a tRNA into the charged tRNA during the process of translation. Now this amino acyl tRNA synthetase, they belongs to the category of ligases. So these are the 6 category of enzymes which are determined by the Enzyme Commission. Now next is the concept of cofactor. The cofactor, simply we can say these are the non-proteinaceous part. The cofactor, these are the non-proteinaceous part of enzymes which is essential for the catalytical activity of the enzymes. The cofactor, they may be of two types, that is the inorganics and organics. Inorganics, they includes the metal ion. For example, like all polymerases, they require magnesium as a cofactor. So the inorganic metal ion, they can be used as a cofactor. Other categories of organics, which mainly includes the soluble vitamins, and these organics, they can be divided, organics except the proteins. So these organics, they can be divided into two groups, that is a prosthetic if the cofactor or the organic molecule, it covalently binds to the enzyme, then it is called as the prosthetic group. And if the cofactor, it loosely binds to the enzyme, then it is called as the coenzyme. By this cofactor, there is a concept of apoenzyme and holoenzyme. The apoenzyme along with the cofactor is known as the holoenzymes. Now, in biological system, as we already stated that there are exceptions. So, there is an exception of enzyme which can use protein as a cofactor. The definition of cofactor is that it is the non-proteinaceous part of enzyme essential for the catalytical activity, but some enzymes, they require protein as a cofactor. Number one is the ribonucleotide reductase, that is the RNR third category. This RNR third, they require activase. Activase is a protein as a cofactor to carry out the conversion of RNA into the DNA, ribonucleotide into the deoxyribonucleotide by removing the oxygen atom from 2' position. So there is a ribonucleotide reductase which require activase protein as a cofactor. Similarly, we are having pyruvate dehydrogenase, which is an important enzyme of linkage step. Linkage step which links the glycolysis to the Krebs cycle. It converts the pyruvate, that is of 3 carbon, into the acetyl coenzyme A, which is of 2 carbon. It carries out the decarboxylation reaction, and this pyruvate dehydrogenase, it requires 5 cofactors, out of which one is lipomide, and this lipomide, it is a protein. So some enzymes, they can use protein as a cofactor. Now, the active site. If the enzyme is made up of around 180 amino acids, then 10 to 20 amino acids, they will form the active site. Now, these 10 to 20 amino acids which are involved in the formation of active site, they are separated by space in the protein primary structure, but they comes together in the secondary and the tertiary structure and forms the all important active site. Now, this active site is the site where there is a binding of substrate. It is the site where the catalysis occurs, where there is a binding of competitive inhibitor जिसके बारे में अपन आगे बात करेंगे, and there is a binding of cofactor which is a non-proteinaceous part of enzyme essential for the catalytical activity. Now, how the substrate it will bind to the active site, and for this we are having two hypotheses. First is the lock and key hypothesis given by Emil Fischer in 1894. Now, as per this lock and key hypothesis, there is a precise compatibility between the substrate and the substrate binding site, that is known as the active site. That's why the substrate binding site, it will readily recognize the substrate. It will act like a lock and key, where the key is the substrate and the lock is the enzyme, and the structure of substrate and the enzyme substrate binding site on the enzyme, that is known as active site, it is same and it will get fit into the active site. But this hypothesis, it was rejected because there is a conformational change which takes place in the substrate after binding to the active site. Now, there is another hypothesis which is called as the induced fit hypothesis given by Daniel Koshland in year 1958. As per this hypothesis, the structure of substrate and the active site of enzyme, it is different. The binding of substrate to the active site, you can see over here, the structure or the structure of substrate and the active site, it is somewhat different, but the binding of this substrate to the active site, it will make the conformational change in both the substrate as well as in the active site in such a way that the substrate will readily recognized by the active site. So this is the induced fit hypothesis which is more acceptable. So this is all about the structure, activation energy, classification and cofactor concepts related to the enzyme. In our next video, we will discuss about the Michael-Menten equation and also about the type of inhibitors. Thank you very much.